everyone, and welcome to today's webinar, Mastering Nucleic Acid Extraction, Principles, Methods, and Troubleshooting. I'm Christy Jewell of LabRoots, and I will be your host for today's event. Today's educational web seminar is presented by LabRoots and brought to you by BioEcho Life Sciences. To learn more about our sponsor, visit bioecho.com. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. The rest of the questions will be answered by email at the address you provided during the registration process. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. Additionally, we would like your further interaction during the webinar. Therefore, we will have several poll questions during the presentation. They will pop up on your screen. We ask you to answer those, and the results will be seen and discussed by our speaker. Now, I'd like to now welcome Laura Torres Benito, Scientific Communications Manager at BioEcho Life Sciences, and she is your moderator today. Laura, over to you.
Thank you, Christy, for the introduction. Welcome, everyone. Before we start the presentation, I'm going to give you a brief overview of the company. Don't worry, it will take just a few minutes, and then I'll hand it over to Nike. BioEcho Life Science was founded on the assumption that even the most routine molecular biology protocols can be improved. And probably you know that the method to isolate DNA and RNA haven't evolved much during the past decades. Therefore, BioEcho developed an innovative technology for nucleic acid extraction, ecolution. Instead of the conventional protocols, which include several binding, washing, and elution steps, with ecolution, the purification is done in one single step. But you will learn more during the presentation. Nike will explain it. This simple workflow brings several benefits. You will shorten time to result, increase quality and quantity of your nucleic acids. You will reduce costs, and you can be also more sustainable. And actually, this is one of the reasons I joined BioEcho, because our green mission is to help scientists become more sustainable in the laboratory. And just to show you one example, this is how much plastic can be saved with ecolution versus a conventional silica-based method. This is an example for 50 reactions of DNA extraction. Well, to finish, just a few words about the company. BioEcho was founded in 2016, and today with around 16 employees. The headquarters is located in Cologne, Germany, and it has a subsidiary also in Boston, USA. And we provide not only a wide portfolio of kits to isolate DNA and RNA, but also tailored R&D services, nucleic acid extraction services, and automation development. If you want more information about the company, products, and/or services, please visit our website or contact us. We will be very happy to help you with your project. And now, without further delay, I'm going to present our speaker today, Nike. So Nike is the Director of Global Support at BioEcho Life Sciences, and she's an experienced biochemist with expertise in nucleic acid isolation and the development of purification kits. She's responsible at BioEcho for providing technical support and exceptional customer service. Welcome, Nike, and thank you for participating in our webinar today. Now you may start your presentation.
Thank you so much for the kind introduction, and also thank you, all of you, joining this webinar. I'm very grateful to be here today, and I already saw that some people we are working with, some of our high-value project partners are here as well. So thank you so much. I feel very honored. As you can see today, I want to guide you through the world of nucleic acid isolation, means RNA and DNA extraction. But as you heard, you rule the webinar today. we prepared some polls, which helps me to understand what you're interested in and what we need to focus on. Feel also invited to discuss with us during the webinar via the chat, via the question chat. There are very talented people behind this chat from my team, and feel invited to chat with them, ask some questions, but also feel invited to discuss with us afterwards. You have all my contact data on the slides. Use them. We love the exchange. So let's start with the first poll. So during the slides or during the presentation, I will disappear from the webcam so you can focus on my slides, but I will appear during the poll to guide you through the questions. So I hope this will pop up now on your screen. So we ask you, why are you here today? Why are you registered for the webinar and what do you expect from us? So I will give you a minute to answer this poll. I hope you all can see it. I hope you're here, for instance, to learn something new, or you're maybe interested in our technology. That would be wonderful. Or maybe just here for discuss something about a sample you are struggling with. All right, so I'm trying now to, I hope you can see the poll. Okay. So I'm trying now to push results to the audience. This is my first time, so I hope you can see something. And I will disappear and move on with my presentation. So, as I mentioned, today it's about to understand how to become a nucleic acid expert. Means understanding the methods which are kind of commercially available, how to use them for different sample types. So usually when I'm starting with DNA and RNA, it's in our daily life. So I like to compare it with noodles. So noodles come in different flavors. They come in different shapes. You also need to cook them differently. And same, but somehow different. And like dealing with DNA and RNA, depending on what you want as an outcome, you need to change the recipe. And also interestingly, if you kind of look a little bit deeper, some of those noodles look like DNA or RNA. Continuing with food, did you know that one of the first DNA molecules was extracted from salmon? So more than 150 years ago, this Swiss-German biochemist discovered a new substance in cells, which he called nuclein. And this biochemist is all but forgotten, but it was Friedrich Miescher who laid the foundation for our daily business dealing with nucleic acids specifically. And he also was the founder of the central dogma of molecular biology. So this dogma states that if we understand a gene, we can also understand its function, means the protein. So it means that if we're understanding the encoding gene, the DNA, and the RNA level, we also understand biology mechanisms. And this helps us for treating diseases, for instance. Misha was not alone. So here are some milestones, but no worries. I don't do kind of a history lesson today, but here are some milestones on the track of RNA and DNA research, which I think are very important to understand where we are at now. So it started in 1866 with the father of genetics, Mendel. I think we all know this still from the textbooks, followed by Miescher, and then also followed by solving the DNA structure by Watson, Crick, and Franklin. And then a lot of other things and findings are going on. So for instance, also there was a fast development of nucleic acid purification techniques, which are still today in use. So there was size exclusion invented, followed by IO exchange. And then in the 80s, the silica method a lot of people are using in the lab was invented. And nowadays, I think all these findings were important because they led us to the invention and launch of, for instance, the COVID vaccination in 2020. But also really miracles are happening when dealing with DNA and RNA extraction, such as cell gene therapy, which makes, for instance, cancer a treatable disease nowadays. So bringing me back Why is studying nucleic acids so important? So as I said, it helps us to understand biology. It helps us to solve a biologic question and also to use this, for instance, in medical treatment. A lot of methods we are using are based by nature, and it's important to understand how nature functions. So nucleic acid extraction is our daily business in life science and life science industries. And to investigate nucleic acid, we usually have a distinct workflow. So probably you're working with different sample types. Maybe it's plant, tissue, liquid biopsy samples, or whatever it is. But usually what you do, you have a huge sample collection, and then you have a huge variety also of samples. Could be blood tubes, could be FFP samples, cell culture. Maybe you want to isolate DNA and RNA or both of them. And afterwards, you are doing famous downstream applications such as sequencing, PCR, could be also microscopy, maybe also kind of checking for genotyping, for maybe doing plant studies and so on. But this all brings us together and combine us to solve a biological question. So no matter if you're doing cancer research or you're working in the pharma industry or even in a molecular breeding, we all want to solve a biology question. And this kind of brings us together because how do we do that? We do this by studying nucleic acids. And this is why we're also here. And the point is that you see also on this slide, you have a lot of things to consider when working with different sample types. And we want you to help you because we all know those days when you're frustrated in the lab and you do not get satisfying results. What we want to help you is to understand that sample quality, so the quality of your sample you get, but also the quality of the eluted sample is the key here. And by focusing on this, we want to help you to avoid this garbage in and garbage out principle we all hear about. So how do we do that? First of all, I think we need to understand the challenges and requirements we are dealing with. So it usually starts with a sample type. So you're getting a sample and you need to understand, what do I do with this sample? And then it's also about, but what I'm interested in of the sample. So means the target. And by having those three pieces, you have maybe to think about the purification method. Do I buy a kit? Do I do it homebrew? What do I do? And this kind of addresses a lot of different questions. So nucleic acid prep is a crucial step for any downstream application. Sometimes it needs to be simple. It needs to fit our downstream application. You want to have high quality. And on the other hand, you want to have something which is rapid, cost efficient, because probably you have a PI or somebody else focusing on budget behind. So you can see here, I try to visualize that everything has to be in a balance so that we kind of figure out where's my balance between all the challenges and requirements so that we kind of fill the circle and make it round and have a successful experimental setup. So before starting to give you an idea about different available DNA and RNA extraction methods, I want to ask you another poll. So I'm popping up again and I'm moving to the poll. So I'm asking you, which method are you currently or mainly using for nucleic acid isolation? Could be phenol, magnetic bead based, maybe also our technology, silica membrane columns, homebrew or direct lysis. So take your time. And I will give you another minute. Oh, fantastic. People are answering. Thank you so much. Okay. Good. Give you some couple of seconds. A lot of people are participating, and it's a very distributed picture here. Wonderful. Now I have the feeling I want to talk to all of you because I'm curious what you're doing. All right. I will display you the results. So it looks like a lot of people are working with kits Some of you are still doing homebrew, so it seems that we have a lot of talented people here, and also direct lysis, so you have a very fast workflow. So I think it's very good because I try to touch a lot of those methods today. If I'm missing something or you have questions specifically, as I said, please feel free to use our team or ask afterwards. Okay, I'm disappearing and continue with my presentation. Yeah, so we know that we have a lot of challenges. We know already, you know, there are a lot of methods around. If I have a new sample, how do I have to start? So where do I start? So usually I try to narrow down everything I'm doing to make it less complicated. I would say if I continue with nucleic acid purification, for me, it's a mission of 2 quests. Quest 1, I have to do the sample extraction. So means I have a tissue piece and I somehow want to get access to the DNA. So I need to release the DNA from the sample. How do I do this? I usually disrupt the sample. If I have my DNA released, let's say, the next quest 2, would be the nucleic acid purification. So my task is here, after releasing the nucleic acid, I need to recover it. I need to separate it from all the other stuff, kind of proteins, membrane debris, and so on. So let's focus first on quest 1, sample extraction. As I said, we want to release the nucleic acid from the sample. So we want to disrupt the sample and then homogenize it. Very crucial step. If you're starting with cells, for instance, sometimes you do not need a real disruption, but we can try to touch this as well. The challenge is here that sometimes people are faced with very hard to lyse tissue or fibrous tissue. Could be bone, could be muscle, but also plant seeds are sometimes not easy to kind of cracked up so that you get access to the DNA. And the point is that nucleic acids, we all know that, are also fragile. If we kind of treat our sample too hard, we risk, for instance, that we completely degrade our RNA. And then the point is that also here, when we try to crush our samples, it could also happen that during this time we are doing this, maybe desperately, that RNA or DNA degrading enzymes are active, like of nucleases, and we want to prevent this. So if we think about sample extraction, it's not that obvious that you're working with one tissue type. As I said, could be hard to lyse, easy to lyse, whatever it is, or different cell types as well. But the goal is always the same. So there are 3 base methods you can use, and you can also combine them. The first one is mechanical lysis. So we have physical methods to extract our sample, to make the DNA RNA accessible. This involves mechanical disruption of the sample to release what we want to have. Often is used this for larger samples, plant material, high cell density. There you have sometimes the tissue rupture, for instance, and complex tissues. Common physical methods include sonication, bead beating, but also what sometimes helps very well is kind of a freeze-thaw cycle, so that you freeze your sample, you kind of thaw it again and freeze it again. This also helps you to crack extracellular membrane structures. Another method is enzymatically. I think you all once deal with lysis buffer having prot K in it. So proteinases are very famous for degrading proteins. We need this obviously because we also have cells with membranes, and on the membranes, you know that there are proteins sitting. And it's getting even worse because also on the DNA, proteins are sitting there. And we want to digest them because we want to have the DNA and not DNA and proteins combined. Then there are also other famous enzymes like lysozyme, thymolase. There's also a collagenase you can use. Enzymatic treatments can be used, for instance, for high throughput processing because they're usually very stable and they come with very low costs compared to other methods. And you can also combine them with mechanical lysis. The next methods I think we all know is chemical lysis. Very easy. So most of the lysis buffers available contain detergents. means soap, basically, because what is soap doing? It lyses membranes. So SDS, for instance. Organic solvents can be used as well. Some people are working with triazole, phenylchloroform, this is what I read, and also charotrophic salts, which also disrupting cell membranes. So we have mechanical, enzymatically, and chemical. We can also combine them. So maybe you can start with the mechanical disruption, powderize your sample, and then add a chemical cocktail, means a lysis buffer, plus an enzyme, a protease, for instance, to homogenize your sample. Usually I'm trying to give you some tips, which you always can go back watching the video again. As I said, I try to divide samples in easy to lyse and hard to lyse. So try to make it easy. For example, easy to lyse are cells, very easy. Exosome, viruses are very easy to lyse. Hard to lyse could be plant tissue, tissue itself, but also insect cells because they have a cell wall, and also gram-positive bacteria or yeast. So there it really helps to maybe add a bead-beating step prior lysis. And there you can also see what properties they have. As I mentioned, the kind of bacteria could have a polysaccharide-rich cell wall, like this mucus around them. And usually that helps to have liquid nitrogen, including to the bead-beating step, to really crack up the whole thing. Also for mechanical lysis, mortar and pestle, means doing it by hand, helps. For instance, when you're interested in really high molecular weight DNA, then it usually helps to do it sensitive. But still, if you have a seed, a plant seed, and you want to get high molecular weight out, you need somehow to do it mechanically with a mortar and pestle, and this helps. So I hope this gives you a little overview about what you can do in Quest 1. So means to extract the sample, make the DNA and RNA accessible. And then what we need to focus on is to kind of distract all the other stuff from the DNA. So means the real nucleic acid purification. This means we have now our sample lyzed, and now we want to recover our nucleic acid pure, sample, flexible, with low cost, contaminant-free, all the things you heard before, and without obviously losing our nucleic acid, because we need yield, because our downstream applications require this. So what are the challenges? I named them before. It's inhibitors, risk of having low yield in the end. And the point is, even if you have an eluate with high concentrated DNA, for instance, if there's still a DNA is active, it would happily digest your DNA when you have your eluate standing around, which is then a pity because you don't have nice PCR signals in the end. So our goal is here to rescue our nucleic acid. There are some methods around. I would divide them in two different parts. On the one hand, liquid-based, for instance, this phenylchloroform method you mentioned before, and on the other hand, solid-based. Liquid-based means that we are working with different liquids and thereby pipetting, let's say, our DNA out. Solid-based means that we have a solid material involved, which helps us to separate our nucleic acid from the other contaminants from our sample. So let's move on. To start with this, I want to move on with the precipitation step. This is a very old method, a very simple approach, and a lot of other methods based on this approach. So you start with your sample, you do your sample extraction. I kind of introduced you before, and we start with kind of a lysate, means the whole mix, proteins, DNA, cell debris in one tube. What you then do is you usually add something which helps us to make the DNA precipitate. This means that this method based on different solubilities. RNA and DNA are usually water-loving molecules and soluble in water. If we want to precipitate them, we add a lot of salt. By that, they kind of getting less hydrophilic. If we add ethanol, so this is the most famous ethanol precipitation, kind of the DNA or the RNA pelleted down. So what you can see in blue, the nice pellet. If we got the DNA pelleted down, we will aspirate all the mix, means the salt and the ethanol, and then we have a nice pellet. We can wash the pellet then and directly resuspend in the liquid we want. This is a very old, but still a very gold method. So people working with plasmid are using this, for instance. And as I said, the principle is solubility. By usage of salt and alcohol, we get our DNA precipitated, and then we can resolve it. This comes obviously with some pros. It's a very rapid and inexpensive method. You got high concentrated sample and also a pure sample. On the other hand, we are dealing with toxic reagents here. So we have ethanol, we can work with carotrophs, and also solubility is always temperature dependent. So means temperature is a factor here. Sometimes it happens that it helps to cool the whole thing down so that you get a nice DNA pellet. And also by pipetting and aspirating, and imagine your DNA is not really precipitated, it means that you can also have a lot of carryovers. So just a risk if you're not familiar with this method. What is cool here in this method that you can kind of play around with the salts you're adding here. For instance, if you want to isolate DNA, you can use sodium acetate. If you have in your lysis buffer STS, it usually helps to use sodium chloride to precipitate your DNA. For RNA, it usually helps to use liquid chloride. Why is that? Why we are using different salts for different nucleic acid types is because they have different solubilities, and we can play around with that. The next method, still liquid-based, is one method you are familiar with, phase separation. The most famous one is phenol chloroform or phenol isopropanol chloroform phase separation. This principle based on solubility as well. So what happens there? Again, you have your sample extracted, you have your kind of lysate with all this stuff, and you want to do a phase separation. What you're going to do is you add your phenol chloroform there, and then usually directly phases kind of turn on. This based on the solubility, again, of DNA in water. I try to kind of explain it a little bit better here. So what you're going to see is that you have, first of all, your lysis means the aqueous solution, and there you have your proteins, and there you have your DNA. Proteins and DNA are usually hydrophilic, so they are soluble in water. With solubility, it means same likes the same. So if kind of a molecule is hydrophilic, it loves water, it stays in water. Now what we need to do, we want to separate the protein from our DNA. We don't want to have proteins in our eluate. So what we're going to do is we add the phenol and mix it. What's happened there is that proteins are very flexible. Usually they don't like to present their outsides in a medium which is kind of toxic for them. Because now we have the phenol outside and the kind of the protein change its conformation and flip its hydrophobic residue outside. So this is why then the protein stays in the phenolic phase here in pink, and the DNA, which still loves water, stays in the hydrophilic phase. So this is what happens here. Sometimes you add chloroform, which helps you to really kind of have a distinction between two phases, because obviously not all substances are the same. There are some proteins which kind of cannot decide whether they want to be aqueous phase or phenolic phase. So this means that they kind of remain in an interface. Chloroform helps here to kind of keep them there so that you do not risk to have protein carryover. So to narrow it down, it's based on solubility and polarity of the proteins and of the DNA, and it's based also on protein stability. So if we have then our aqueous phase, what people are usually doing, you kind of aspirate the phase, and usually people are proceeding with a precipitation step or with kind of maybe also a column step. So just that you have an idea. So usually the phase separation is kind of a prior step before precipitation. What are the benefits? I mean, a lot of people are using this, so there must be benefits. It's low cost. So usually it's quite low cost and you can use a lot of input. It also gives you kind of a very good yield. And you can do RNA and DNA as well. Why is that? Because DNA and RNA have different solubilities. If you play around with the pH, you can somehow enrich DNA, but also enrich RNA. And this is sometimes very helpful. You can also, and there's a substance around which is called trisol, you can also isolate all the three substances from one sample by using this different pH shift. There are some disadvantages. This method is very toxic. So be aware, kind of a word of caution for you. Phenol is a toxic chemical. It's poisonous and corrosive. So if it comes to your gloves, please take off your gloves quickly because it's really important that you take care here because it gets into your skin. There's usually no automation possible. It's not easy to automate phase separation. And there's still a risk of contamination because let's say the following. If you are too kind of brave aspirating the upper phase, you can disrupt the lower phase and kind of have a phenol carryover. So you transfer the phenol in your alouette. And this will completely ruin your PCR. What also can happen if you're too timid, you have a low yield because you leave a lot of aqueous phase in the sample and don't use it for downstream. And in the end, you do not have high yield. So just to give you an idea that it's very important that you be careful during phase separation. And next step is gel filtration. So there is a nice method based on hydrodynamics where you kind of can apply your lysate on a column. And then with the right running material, it kind of flows through the column. This is usually combined with anion exchange for high molecular weight. But there's some famous gel filtration around. You all know this, kind of a agarose gel. So there it's also used this method. So you apply your DNA molecule and based on the size, it flows through the column material or the material, the gel material you have. So just to give you an idea how this works. And usually it's just by gravity, or in case of gel electrophoresis, it's by electricity. This is a also very flexible and rapid method because, I mean, you have your lysate, you apply it, and then it kind of get distributed and you see your DNA fragment and get it eluted by size. However, it's limited to volume. We all know this. If we load a gel, you usually have just a tiny volume you can load. And it sometimes happens that your DNA stays somewhere but does not run anywhere, or it kind of disappeared because it ran out of the gel. And this is also kind of the worst part here, that if you apply something, it's not given that it runs through the column. And also there could be a risk of contamination because it's by size. We all know this, ugly gels, when you have kind of another band or maybe salts in your band, it's not easy to isolate a nice DNA fragment after PCR. Now coming to something super famous, I think you're familiar with columns. Could be columns, could be also plates. But what I want to introduce you is the solid base nucleic acid extraction. Means we have a solid material which interacts with our DNA. So we lysed our sample, we have our sample lysis, and now we bind it to a column. And my example, it would be a silica column, which is the most famous one. So it has a little silica membrane in the column, which helps us to get our DNA rescued. And this process based on the bind, wash, elute workflow. So DNA binds to the matrix, not just because for fun. Usually what we are using in our binding buffer, it's caltrophs and also ethanol, which helps us to kind of bind the DNA to the column, means make it unsoluble for a second, and then somehow to rescue it in the end. So what we're going to do is we mix our lysate with caltrophs and with ethanol, and then we apply it on our matrix, spin it down, and then we have to wash the membrane. So keep the DNA, hopefully wash other stuff away, and then we elute our DNA in a lower volume. So you can see it here. It's DNA binding, washing, and elution. examples here are not only the silica membrane, but also the ion exchange. And because I think a lot of people are using this method, I want to show you how this is really working. So usually what happens is you see here on the left the silica membrane. So with this SEOH groups. If a carotrophs comes to the membrane and touch it, what happens there, including salt here, means not sodium chloride, there's kind of a salt bridge building up. So means you see now that the sodium ions are binding to the oxygen. So when then now DNA kind of is phased to this membrane, there's an ionics exchange between the sodium ions and thereby building a real tight bridge between the membrane over the sodium to the DNA. And this is just because of different charges, which means it's kind of a stable bridge, but you saw we apply salt, this bridge built up. If we reduce the salt content, the bridge will kind of get broke down again, and then we can rescue our DNA. So this is really what happens during this method. Why people are using this? People are using this because you get high pure samples. People are using this because it's easy to handle. You know what you're going to do. There are commercially available kits for this with nice instructions. It's very well established. A lot of people are using those methods, different providers, but there are some kind of disadvantages. It's input dependent, obviously, because if I bind something, sometimes the binding capacity is done. So means we need to figure out how much we really can bind to this membrane. And then we have a lot of chemical waste because carotrophs, ethanol, for all the substances, you have to do a different lab waste management. And then there are costs because it's not only the lab waste management, but buying a kit sometimes could be expensive. So just keep this in mind that it's a very good method, but it also comes with high cost and also with chemical waste. Just a tip from my side, a lot of people are looking for microRNA as well. If you are faced with the study, you can, for instance, increase the amount of alcohol because the alcohol here helps us to interact or to let the DNA interact with the membrane. If I increase the amount of alcohol, I usually bind, let's say, smaller fragments, which microRNA definitely is. If you have questions to that, please feel free to share them with us. Well, we have other solids DNA and RNA can bind to, and those are magnetic bead-based kits. So means that you cannot only work with columns, you can also work with particles and like called magnets or super paramagnetic particles, which are little micrometer-sized balls in a suspension, which helps you to recover your DNA. This works similar to the silica approach I showed you before, but there could be also pH-dependent beads so that you're working with shifting the pH to bind the DNA and also to release it. So usually what you're going to do, you have your lysate, you add your beads, and then you mix it gentle. What the beads are usually doing, they interact with the DNA molecules in this case, whereas proteins, for instance, should not interact with the beads. You then apply your magnet. So means that you have kind of a condense beads on the side of one tube, and the DNA molecules are still bound to the beads. Then you have in your supernatant, hopefully, impurities, proteins, cell debris, which you can then easily wash away. And then with kind of releasing the magnet, applying the magnet, and adding wash buffers and so on, you somehow get to your pure alouette. This is also a very convenient method. It also based, as you probably realized, on the bind wash elute workflow. And it based also an affinity purification, means this interaction. The DNA molecule interacts with the beads, similar to silica membrane. It definitely comes with some advantages. It's very easy to automate. Maybe you also have an automate in your lab. So it means a device which has a magnetic rack, and then you apply your beads, and then you do a magnetic bead purification. It's flexible to input, and usually it prevents your method from clogging, because usually if you have a lot of input, you can adapt the amount of beads you're using. There are some disadvantages. If I have kind of a high input, I usually try to increase the amount of beats so that I catch a lot of DNA molecules. The point is that it can come to the point that once the magnet is not able to handle all the beats, which means that you risk of having beats in your alloy. Having beats in your alloy will kill your downstream applications. So if you're working with magnetic beats, be aware of having the right beat input ratio. It's also important that those beads are sensitive to viscous samples. So if you're having, for instance, a lysis buffer which is getting viscous by temperature, you need to pay attention that this buffer has the right temperature so you can use it for for magnetic bead-based purification. And then what is also important is easy to automate, but it's kind of tedious for manual handling because you always have to apply the magnet and release it. So I hope this gives you an idea about the methods which are commercially available. There's obviously something like kind of direct lysis, which some people of you mentioned before. This means we lyse our sample and we take directly from this sample a little amount of volume and can go to our PCR, which is also a method, works very well and very fast, but sometimes it kind of comes to fallout rate. Why we are here today and why we are talking about this. Laura mentioned this briefly. We believe in BioEcho that there must be always kind of new inventions in life science. And we saw this with downstream applications. I mean, imagine the following. I mean, you can sequence your whole genome for less than $1,000. It's mind-blowing. The point about nucleic acid purification is that you saw this probably before. It was invented in the 80s. So all the methods I showed you before are quite old. And if we think about the 80s and the 90s, it was so cool to have such a huge video camcorder going out and kind of film whatever you see. But now, I mean, we all have a little computer in our pockets, a little smartphone. But interestingly, nothing changed on the DNA and RNA extraction methods. And this is somehow kind of puzzling us. So, and we think in our world, it's not the purification methods rules here. It's the application method, so means downstream applications. And the point is that we see that coming maybe to sequencing, there was a dramatical invention. It started with single analysis, parallel analysis, and now we can do a lot of sequencing approaches in one experiment, and also multiplex PCR. But nothing has changed in the sample preparation. So means how should all the downstream applications handle it if nothing kind of is improving on the sample prep field? And this is what we're gonna kind of want to change. We want to kind of fill the gap between sample prep and downstream application with our technology. Because we have something which probably is new to you, but which completely fits to the downstream application requirements. We have fast methods, it's simply, and it's also scalable. And for us, very important, it's also sustainable. So we get rid of those toxic chemicals I mentioned before. How does this technology work? So we can work with several different input types, FFPE, cell, RNA, blood, plant tissue, whatever it is. The whole workflow is the same. So we first of all, as I mentioned, have to do quest one. We have to do our sample extraction. We have to make our DNAs accessible. So we have our lysate, which you can see in this drop, with cell Jabry's nucleic acid. And then we apply this lysate on our membrane directly and proceed in quest two. With a single step purification, we elute our pure nucleic acid, whereas the other stuff remains on the matrix. And this makes us faster and also more sustainable. I explain you how. So here you see for an example, the spin column based method I presented you before, the bind wash elute workflow. Just to recap, we lyse our sample, we bind it to the matrix, we wash it, and then we elute it in a smaller volume. And sometimes this could be that we have several washing steps depending on the sample. Switching to our technology, we were able to combine the bind wash elute workflow in a single step purification. And thereby getting not only faster, but for us more important, We also save plastics because we're saving 1, 2, 3 pipette tips, binding, washing, elution. And also by not having a washing buffer, by not having a binding buffer, and by not having a real elution buffer, we completely save liquid waste. And we're also following a sustainable, greener chemistry project. So we try to get rid of all the hazardous chemicals. So on this nice graphs on the left, you can see that kind of using or switching to our methods, you dramatically reduce liquid waste, which is very important to us. So to narrow it down, I think the kind of famous conventional methods around are the silica membrane-based technology I showed you before, then the magnetic bead-based technology, and then also the technology we have. The silica membrane, for instance, could be always combined with the phase separation methods, means the phenochloroform, which a lot of people are doing. But usually people end up with kind of a column or bead-based kit. So why we are here? Because sometimes life is not easy when dealing with samples and DNA and RNA extraction. We all know that life could be nice in lab. We all know that there are a lot of days where it's not. So we believe that if we improve the sample prep and extraction, you will kind of get successful. What could happen if we kind of have struggling with our preparation? Obviously, if we do not choose the right method, we have a drop in performance. We have maybe ethanol carryover, which kind of disrupt our PCR. We have to do it again, which increased of our costs. More important, we drop in satisfaction. I mean, I remember the days where I kind of desperately isolated RNA and kind of get to my bioanalyzer data and where I was always disappointed, which kind of drives me nuts. And also very important, delays in project, which sometimes it's not nice working in lab and a PI or kind of a manager is asking for results and you cannot deliver. And as I mentioned, what is important here, we need to understand that all these points kind of tell us that our balance, the demands on our sample extraction is kind of disrupted. So imagine the following. We know how to deal with our DNA and RNA extraction. We are very imbalanced. But then a new weird sample is coming. Could be a beetle. Insects are not easy to homogenize. FFPE, we never got in touch with FFPE, or blood, coming with a lot of proteins, and it's not easy to isolate DNA or RNA out of it. What happens there? It could happen that our balance got imbalanced, and then we kind of fail in our DNA and RNA extraction. So I have a question for you. What do you think might affect the quality of your DNA or RNA sample the most? Is it process time? Or maybe age of the sample? Nucleases? Or those inhibitors which can somehow appear? Some people are already answering. Thank you so much for participating. It's very interesting. So cool. I give you some couple of seconds. I highly appreciate it. I really like it. It's my first time to using those polls, and I really love it. Good. So I will try to show you the results. Here we go. And what you can see here, that it's not really clear what kind of affects the quality. For some of you, it's process time. For the others, you are probably dealing with wrong stored or kind of old samples. And some of you have issues with RNA or DNA degradation, which means this active residual nucleases. And yeah, inhibitors are usually a huge problem when dealing with plant material, tissue material. yeah, it's definitely sometimes user handling, but honestly, we are all scientists, so I think we know what we are doing. But yeah, we can definitely pay attention on this. But I have to say, usually if you plan your experiment right, please don't judge yourself so much. It's not you. Usually it comes from the sample and we just need to find the right method. So don't frustrate yourself so much. Good, I'm moving on with my slides. So we know there are a lot of sources of kind of stuff affecting our DNA and RNA quality. What to focus on? So you named some points, I think, which are very important. And coming back to our circle, which we want kind of to keep close and round. On the sample side, you're completely right. It could be a complex sample, FFPE, super complex, coming with a lot of different substances besides our tissue. It could be age, storage. If you have a tissue kind of waiting outside on room temperature for hours, kind of the chance quite low that you get nice RNA values out of it. And then it also nucleic acid content. There are some tissues coming from nature with low RNA content. And we need to know this before starting because otherwise we're getting frustrated. Then it's also important to think about your target. Am I dealing with gDNA? Am I dealing with RNA? Because RNA is quite unstable compared to DNA. Do I want to have microRNA or mRNA, or I'm interested in plasmids? And then obviously what also affecting is the application. Why I'm naming the application? Because the application comes last. The application usually rules if we had success or not. Because I mean, if you're doing your DNA isolation, have nice integrity, but your kind of sequencing facility is saying you, yeah, but the concentration is too low. I mean, you have still a nice DNA extraction, but the concentration is too low, so you cannot move forward with this. So keep an eye on your application. What is needed there? Because if this application is not sensitive to kind of absorption ratios, it's fine, move on. I mean, you do not have to have perfect values if you do not need them. And based on this, you can then choose your purification method. You can choose direct lysis or the other methods I kind of presented you. You can also choose always between homebrew and commercial kit. So again, what to focus on? Focus on your downstream analysis. Understand your downstream analysis first, and then move on and think about what you need. Think about the specs and requirements. What yield do I need? How pure need my sample to be? And what integrity do I need? Here you see some examples of our internal data. So you can see that we are getting good yield and pure samples and also integrity, but it not always have to need to be like this. But also important, you mentioned it before, think about your influencers. Think about handling. How do I handle my sample? And this is also where I think storage is a huge impact. Think about your sample itself and the purification method you're choosing. So know your troublemakers. Usually I try to narrow this down in 4 points. The sample type means how is the sample collected and which state the sample is, sample disruption, sample lysis, and DNA and RNA isolation. If we start with the sample type, it's definitely the age, maybe the DNA and RNA is already degraded. Maybe there are a lot of polysaccharides binding to our DNA and kind of get co-eluted and the sample is then not pure, or we have cell walls. There we can think about the input amount if the DNA is degraded and we try to get something out there. We can think about to improve the storage. We can think about additives, which helps us to prevent, for instance, binding polysaccharides to DNA, and stabilizers, RNA or something like that. When it comes to sample disruption, we need to think about the method we are choosing. If we are too hard, we degrade our DNA or RNA. If we are too soft, we do not lyse our sample anyway. And also beads are usually very important. If I'm choosing the right bead material, I can win a lot here. And then it's also important to think about, do I need liquid or make it sense to disrupt with liquid nitrogen? For plant seeds, for bacteria, this is very beneficial. The other second points are sample lysis. So sample lysis can be based, as I said, on chemicals, but also with the help of enzymes. Enzymes usually love to have heat because then they react faster. Then you have also to think about binding. What happens if something binds to my enzyme and completely inhibit it? I won't kind of get the enzyme digestion successfully done. So it means I have in the end a lot of nucleases active in my aluate, and those will degrade my DNA or RNA. And then what happens as well is, as I mentioned before, if my sample disruption was not right, I still have intact cells, which kind of not get fully disrupted, so means my yield will be low. Afterwards, if I'm done with sample lysis, I still have the point that I have my DNA, but I want to get it separated from the other stuff. Polysaccharides, proteins, active nucleases, you may name it. What could also happen, what you can see with the histone, that the DNA is still bound to proteins, which means that it could be that my DNA is even not binding to my magnetic bead, or it does not get eluded. So means I cannot use it. So it is very important to think about it. I do not have yield. What happened here? Is it because of this sample disruption, about the sample lysis, or it's because I did not even get DNA eluded? What helps here, for instance, if you have a flow through or something, to check your flow through if your DNA is there, because this means that DNA was not able to bind to your material. To give you some examples, I want to give you 3 little studies. First of all, about yield. Here on the left, you can see in gray, usually the standard profile, nice nanodrop data, nice PCR data. If I have low yield, usually I have a drop in the nanodrop and also a shift in CT values in my PCR. What could be reasons? For cells, it could be that you have a frozen pellet, but somehow it's thawed and means, or it was not resuspended gently. So it means that I did not really mix it with lysis buffer and I still have intact cells which get not disrupted. So it means my yield is low. Could be also that I have too much input, everything is getting viscous, and the enzymes do not like viscosity, so they do not do their enzymatic step right. Or we even elute successfully DNA, but we have still active DNAs in my aluate and completely degrading our DNA. For blood, it's same. If blood was frozen and kind of heat up too quickly, it could be that the residual nucleases are still active and degrade our DNA. Blood comes with a lot of proteins. It sometimes happens that there is kind of a co-precipitation of hemoglobin. So it could be also that your membrane gets brownish when you add your blood to your silica membrane, for instance. This is the hemoglobin. It's fine if it's there, but it sometimes happens that it gets co-eluted and then it inhibits downstream. For tissue, for instance, you named it before, storage is important. If I store my sample wrong, if I disrupt it wrong, I do not get yield. If I have too much input for columns, I will clog them. I can overload also my beads. It could sometimes happen that kind of beads get stuck between proteins and I don't win here. So input is also important. If you work with a new tissue, do kind of a dilution. Try different input amounts. For purity, it's kind of similar. Here you can see kind of in gray again, high quality nanodrop graph. And in red, you see kind of a phenol shift. So we have a phenol carryover, which means that my absorption curve is kind of disrupted and I cannot determine my right yield. You can see, for instance, in PCR that contaminations usually affect PCR results as well. And this then result in higher CT values in this case, which you can see, for instance, in gray. What is important here? It's important to focus on handling. If our sample is super old, it could be that we have kind of degraded nucleic acid. If I'm working with plant material, for instance, it's coming with a lot of secondary metabolites. Contaminations of RNases, for instance, does not help us as well. Or what is also important, expiration date of kits, and also kind of carryover of ethanol. Expiration of kits is very important. If your kit is expired, it could sometimes happen that the lysis buffer, for instance, it's not active anymore. And this means that our lysis is not effective, which leads also not only to low yield, but more important, low purity. So if you have low pure samples, check the expiration date of your kits. Very important, if you're dealing with ethanol, always make the ethanol fresh, because ethanol usually kind of contains water very quickly. So if you start with a 70% solution, it could be that it's already at 40% after a week. Other samples could, as I mentioned, contain a lot of nucleases, so we need to be aware of this and study our sample before starting with this, or polysaccharides, protein nucleic acid complex. The purification method is here crucial. As I mentioned, it could happen that you have ethanol keratose carryover. Could mention that if you have fibrous tissue, that you have clogging. That's also important if you're using RNA later. It also sometimes happens that your tissue got kind of fibrous, so it's got kind of gluish. So there it's kind of helping if you do a sample disruption. And then it's also important to think about what could impair my binding. Everything was similar to DNA impairs your binding, for instance. Last but not least, integrity. Dealing with sequencing downstream, probably you were all faced with integrity ones. People are saying, yeah, but what's your DIN? What's your RIN number? And so on. So usually we kind of investigate integrity via bioanalyzer or via tape station. Here you can see some results on DV200 and tape station data. What does DV200 mean? DV200 is kind of an alternative for RIN values. It shows you the percentage of the fragments which are bigger than 200 base pairs, so that you can have an idea how much fragments are bigger than 200 base pairs. There you can also see a tape station graph from an RNA isolation, which shows you the ribosomal peaks, which gives you also an indicator about how integer and stable my sample is. What affect integrity? Yeah, the main things as before. Age, kind of how I handle the sample. Temperature, obviously, as I said, keeping the tissue on room temperature. Active nucleases, which completely degrade RNA if they're present. The kit, obviously, but also homogenization. If I homogenize beat my sample too hard, I will degrade my DNA or my RNA before even starting with my purification. Binding is also important. Beads and also silica membranes are kind of selective to the fragments they are binding. So usually the cutoff for silica membranes is around 500 base pairs if I do not adapt the binding conditions. And for beads, it could be somehow around 200. So this is also important. If you have the feeling that you kind of get not the fragment you want, check on the method you are using. So I came up with a lot of stuff today. As I said, you can always go back and check the webinar out. To narrow it down, I want to give you kind of 4 important questions to understand how sample, target, application, and purification method is related to each other, and how do I get nicely my DNA and RNA out. It's what type of sample and how much am I using. So study your sample and think about how much do I need based on your application method, what your application is needing. Which type of NA, of nucleic acid, is needed? Do I need the whole RNA, or am I just interested in smaller RNAs, 200 base pairs or something? For what am I doing this? What is my aim? Because this is important, as I pointed out. If I'm not interested in absorption ratios, I should not care about them. If I'm interested in kind of high DIN values, I should focus on that. And how can this be achieved? And then choose your purification method, not the other way around, taking just a method and desperately looking for good results, because this leads to you to frustration. Yeah, and with this, I want to end up. I can just say, be a nucleic acid expert. This is important to me. Feel confident about what you're doing, because usually it's not you, it's the sample and the method. And thank you so much for your attention. I can say we are at the beginning of our journey. If you never heard about our technology, feel free to join us and on our journey and test it out. And now I want to go with Laura through some of your questions and answer them.
Fantastic, Nike. Thank you for your insightful presentation. I'm sure that our participants have learned a lot about nucleic acid extraction. I really like it how you picture the whole procedure as two mission quest. Okay, now we will now start with the live Q&A. So if you have some additional question, many people already submit their question. You can click in the ask a question box, and then we will answer as many questions as we have time for. So, Nick, the first question. Someone asked that this person is performing transcriptomic analysis, and so is wondering how the quality of the RNA can affect qPCR and the sequencing results.
Mm-hmm. Okay, very good question because it covered a lot of downstream applications. So the quality of RNA is crucial. For sequencing, obviously, what do you need? You need concentration and you need a good RIN, means integer samples, so means big fragments, so that you get in the end nice sequencing results and understand what the sequence is. So for PCR, it's not that crucial to have such long fragments depending on what you're going to see. And what you want to see means how big your amplificate is. And this means now, how does it kind of affect then the downstream applications? If the RIN number is very low for sequencing, you do not get much reads. It could be that you only sequence a little piece of your RNA, and then you have to puzzle because then you get probably a piece here, a piece there, and maybe you're missing the piece in between. So it's not easy to analyze this. So the better the RIN means kind of the longer the fragments are you can get. I hope this answered the question. What do you think? Anything open from your side?
Yeah, sure. I think that for qPCR is not that critical, but for sequencing, you really need high quality and
Definitely.
integrity. Yeah.
Maybe
Absolutely.
maybe one point, which I'm thinking now about, what could affect as well is if we have contaminants there coming maybe from our sample or coming from our purification method. This is crucial. If you have phenol carryover, for instance, they need to get rid of this. For instance, for PCR, but also for sequencing. What is not good is if you have active nucleases there. Imagine the following: you have your RNA sample, and then you start to do your polymerase. So means your RT reaction, making of the DNA, ctDNA again. And you have kind of an active DNA sitting there and waiting for the ctDNA and kind of digest it again. You do not get anything in your PCR, and this is kind of
right.
crap.
Yeah. Thank you, Nike. The next question is about yield and concentration. They ask if you have a very small amount of material, and this material is very important, if evolution have a step of volume reduction.
So I do not know what sample type you are referring to. So what we can do, so BioEcho, maybe I go back to the slides. Maybe I make it easy for you see. Let me go to the technology. Okay, hope you can see the slide here. So what we are doing with BioEcho, the volume we apply on our matrix, it flows through a matrix. Means that our matrix is designed to a specific amount of volume. So if we apply, usually we apply 100 microliter, and we also get eluted in 100 microliter. If you have if we can narrow your lysis down in 100 microliter, that's fine. What we also can do, we have little smaller columns. So if we switch to a higher format, like a 384 well plate, there we can work with 25 microliter. Maybe we can discuss this further, but what we do not have BioEcho is kind of the thing with bind wash elute workflow, the up concentration effect. Maybe to explain it in the upper part of the figure, you have a huge volume of lysate here. You apply it on the matrix. And then you can see we elute in a quite smaller volume. This is an up concentration effect. This based on that the DNA interact with the matrix, and then I can elute it in a smaller volume. This is what echolution does not do. There's no interaction, which means also we do not can kind of affect the elution volume because we elute everything we apply. I hope this answers the question.
Mm-hmm. And I think here it's also important to think what do you need for your downstream application, if you need more concentration or yield, because that's different, right?
And maybe to the person who was asking the question, I'm very interested, because if you have a liquid sample or do you have a tissue? Because what we definitely can do, if you have, for instance, a little piece of tissue, we already had this little tiny one, we kind of adapted the lysis volume and then it worked for us. So don't give up here. Reach out to me if I can help you if you want to.
Thank you, Nike. Next question is about automation. So it's a very specific one. So the question said, can you automate your process and what is the maximum throughput achievable? Samples per hour, they are asking, specifically interested in DNA from blood for high throughput sequencing. So yeah, very specific question.
Okay, fantastic. I like it when it's specific. Yes, we can. Why can we do this? Because it's kind of a column-based method. You can automate it. What you can start with is a liquid handler, because it's then just transferring liquid, kind of a semi-automation, and then you take your plates out and put it in a centrifuge. If you don't want to do this, there are also liquid handlers. We are working, for instance, together with Hamilton. They have a centrifuge on deck, so you have everything on deck. Depending if you have it on deck, you can process less samples than if you have kind of just the liquid handling on the deck. Also depends on the protocol. We calculated for different products. What can I say is that we are indeed able to do more than 3,000 samples in less than 4 hours on a liquid handling system, fully automated. So if you need help there and interested, please get back to us, because I think, that would be interesting also to hear your blood approach.
And we have a fantastic automation team.
Oh yeah, we have. They helped me out a lot.
Nike, you talk a lot about challenging samples during your presentation. And I have a question about FFPE, which are such a valuable samples, but really difficult to isolate. So how can someone prevent extraction failures from FFPE samples?
I think, so yeah, FFPE is a very precious sample. The question is, what do you mean with extraction failures? Because usually people, when working with RNA, for instance, they expect from the textbook high RIN values. And I think that's not feasible with FFPE, because how the FFPE sample is handled completely kind of ruin our RNA or RIN values, because it's cooked completely before embedding it in paraffin. So what you can focus on, and I touched this briefly, is DV200 values. So the left graph here. So means not focus on RIN, but check on your tape station. You can do this manually. There's a nice kind of instruction from Agilent. Check for the fragments which are higher than 200 base pairs. This helps you to understand if your sample is still integer. And you can still sequence your samples if it's around 40%. So what I can say is that if you have the feeling that RIN number is quite low, check on DV200. And what I also can say with FFPE, it's important to think about the input, because usually what is the worst part of the FFPE samples is the paraffin. We want the tissue. We don't want the surrounding paraffin. So if you have the chance, reduce the paraffin as much as you can, trim it, because by that you put a lot of tissue in the sample and thereby kind of preventing paraffin carryover. This usually helps. And then it's just isolating tissue with some kind of specialties.
Thank you, Nike. Yeah, tricky samples. So, Nike, thank you very much. I would love to be discussing with you here for really long time, but I think it's nice if we conclude our session today. But do you have any final comments for our audience? Some
Of course.
final tip?
Yeah. Yeah. First of all, I see all your nice questions, and I think a lot of them are answered, but we definitely will follow up on your questions. So if they're not completely answered, please be patient. We will answer all of them. I would love to take all of your questions and discuss with you. And what I want to say is thank you so much for joining us today. And very important, if you want to do those DNA RNA work, focus on what you need before starting an experiment. Plan it right, kind of do some research on your sample, because you are important here. You are the brain here. Don't frustrate yourself. Make a good experimental design, and then you will have success.
Thank you, Nike, for the nice session. And yeah, we hope you enjoyed the webinar today and hope to see you on our next webinar. I hand to Christy. Would you like to do the closing, Christy? Thank you.
Laura and Nike, thank you both so much. I would love to close us out. Before we go, I do want to thank our audience for joining us today and for their interesting questions. To reiterate, questions we did not have time today and those that are submitted during the on-demand period, they will be addressed by our speaker via the contact information you provided at the time of registration. Today's webcast can be viewed on demand, and Laborats will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Now, at the end of our presentation, a survey will pop up. You can also find it on the far left of your screen. We're grateful for the time you spend answering those questions. It does help us learn that if we covered your expectations, and it helps us improve your experience in our upcoming webinars. That's all for today. We thank you so much for joining us. Everyone have a wonderful day. Bye-bye.
Bye.
Bye.